Good afternoon everyone. Today we will be dealing with anatomy and physiology of cornea. As we all know, the cornea is transparent and avascular structure. It forms anterior 1/6 of outer fibrous coat of the eyeball. The average horizontal diameter of cornea is 12.6 mm. Average vertical diameter is 11.7 mm. Central thickness of cornea is 0.5 mm. Peripheral thickness is 0.67 mm. Radius of curvature of anterior surface of cornea is 7.8 mm and posterior surface is 6.5 mm. So cornea is prolate shaped. That means it is flat at the periphery and steeper centrally. The refractive power of cornea is 43 diopter. That means it forms 3/4 of total refractive power of the eye, which is 60 diopter. Refractive index of cornea is 1.376. Now we'll see the layers of cornea. As we can see in this picture, there are 5 layers of cornea: epithelium, Bowman's layer, stroma, Descemet's membrane and endothelium. There is one more layer that is called as pre-Descemet's membrane or Dua's layer. First we'll see the corneal epithelium. Corneal epithelium is embryologically derived from surface ectoderm at around 5 to 6 weeks of gestation. It is a non-keratinized, non-secretory, stratified squamous epithelium. It measures around 40 to 50 micrometer in thickness and it consists of 3 types of cells. The innermost is basal cells, middle one is wing cell or suprabasal cells and outermost is the superficial cells. The basal cells, it is a single layer of columnar epithelial cells. It originates from stem cell of basal layer of limbal epithelium and they are distinguished from the limbal stem cell by expression of keratin pair K12 and K3. It is capable of mitosis. It is source of wing cell and superficial cells. It contains more mitochondria, endoplasmic reticulum, Golgi apparatus which stores the glycogen. It also forms store of glycogen. Basal cells are attached to the underlying basement membrane by extensive basal hemidesmosomal system which are integral membrane protein complexes. The lateral intercellular junctions have gap junctions. In this picture we can see the tear film followed by surface of flat cells, then polygonal cells, the basal layer which is single columnar layer followed by basement membrane. The wing cells, these are 2 to 3 layers of wing shaped cells. These are less flat cells containing abundant intracellular keratin tonofilament. They have similar intercellular junction with increased in protein ZON when superficial cells are damaged. The superficial cells, these are 2 to 3 layers of flat polygonal cells with extensive microvilli and microplicae. These microvilli are covered with fine closely opposed glycocalycial layer which contains transmembrane mucin 1 and 4 protein. These apical villi, they increase the surface area of contact and adherence between the tear film mucinous undercoat and glycocalycial cell membrane. The zonula occludens seen in the lateral walls are found in this layer only. Here we can see the tear film and the microvilli which attach to the tear film with the help of glycocalyx. And here we can see the layers: superficial layer, wing cell layer, basal layer, and which is attached to the basement membrane by desmosomes and hemidesmosomes. The basement membrane, the basal cells of epithelium rest on the basement membrane. It has major component of type 4 and 6 and 7 collagen. Also it also contains laminin, fibronectin, fibrin, percalin and heparin surface. Now the second most important layer is Bowman's layer. It is the anterior condensation of substantia propria, that is stroma. It contains type 1 collagen. It is approximately 12 micrometer in thickness. Once eroded, it does not regenerate, so it is not a true basement membrane. The third layer is stroma. It makes 90% of corneal thickness. It constitutes mostly type 1 collagen with lesser amount of type 5 and type 6 collagen. The stromal collagen fibers are arranged in sets of lamellae which are approximately 200 to 250 in numbers. They lie parallel to the corneal surface and at angle to the adjacent lamellae. The interfibrillar spacing of less than a wavelength of light, so it does not scatter the light, allow the transmission of more than 99% of incident visible radiations. which accounts for transparency of the cornea. The fourth important layer is Descemet's membrane. It is a true basement membrane secreted by endothelium throughout the life. It is approximately 3 micrometer at birth, but thickness increases to 10 micrometer with the age. It consists of collagen fibrils, which is bound in a matrix of glycoprotein. At periphery, it appears to end in an anterior limit of trabecular meshwork, which is known as Schall baseline. And on electron microscopy, it is divided into two distinct regions: anterior 1/3 that have vertically banded pattern, and posterior 2/3 which appears amorphous and granular. It is highly resistant to proteolytic enzymes, hence Descemet's membrane may remain intact even in case of several corneal ulceration, forming a descemetocele after destruction of epithelium and stroma. So it is very resistant to any chemical injury. Its warts-like excretions in the periphery seen in old ages, which is known as Hassel-Henle bodies, and the central excretions are pathological, which are called as guttata. These are seen in case of fused dystrophy. Endothelium. It is a monolayer of endothelial cells having hexagonal in shape. The basal surface has numerous hemidesmosomes which promote the adhesion to the Descemet's membrane. These cells have gap and tight junctions along their lateral borders that does not encircle the cells completely, hence called as macula occludens. This forms a leaky barrier between the aqueous and stroma. Here we can see the polygonal shaped endothelial cells. The cells contains large nucleus, and endothelium is rich in mitochondria and Golgi apparatus and endoplasmic reticulum. There are minimum 400 to 700 cells required for normal corneal function. The cell density decreases with the age. The central endothelial cell density decreases at an average rate of 0.6 per year, and the normal endothelial cell count is 4000 to 7000. Now we'll see the corneal nutrition and metabolism. The corneal metabolism depends on oxygen, predominantly from the atmosphere and also from aqueous humor and limbal vasculature. During sleep, oxygen from the highly vascularized superior palpebral conjunctiva, and high corneal metabolism is maintained by corneal innervation in the epithelium than stroma. That means epithelium plays important role compared to stroma in maintaining the corneal metabolism. The metabolic requirement of glucose, amino acid, vitamins, it is supplied by aqueous humor through ciliary body, tear and via limbal vasculature. The glucose additionally from the glycogen stores in epithelium. In cases of wound, any injury to the cornea, there is property of endothelial wound healing. Endothelial cells, these are non-replicating, that is, it does not degenerate in the human beings. So damaged cells are rapidly replaced by spreading and enlargement of adjacent cells and their centripetal migration into the injured region. Cells as far as 250 micron from the wound edge are involved in migration. The cells near the wound edge migrate at 800 to 100 micron per day at initial stage of healing and then spread to fill up the injured area. Then remodeling of cells into more normal hexagonal shape occurs. Then pump and barrier function of the endothelium is re-established and cornea returns to its normal thickness. Now we will see the stromal wound healing. Stromal wound healing involves resynthesis and cross-linking of collagen, alteration of proteoglycan synthesis, gradual wound remodeling, and the stromal response to injury is directed towards the restoration of tensile strength. So following injury, activation of stromal keratocytes undergo process of transformation, that is hypertrophy and proliferation, which enlarge in size within 6 hours and migrate into injured area within 24 hours. It undergo apoptosis and activates the adjacent keratocytes, and within 1 to 2 weeks myofibroblasts enter the area of wound. So remodeling of extracellular matrix occurs. Now the most important aspect of cornea is corneal transparency. The cornea is transparent because of following factors: First is corneal epithelium. Normal epithelium is transparent due to the homogeneity of its refractive index. The basal cells are firmly joined laterally to other basal cells and anteriorly to wing cells by desmosomes and macula occludens. This tight intercellular junction accounts for epithelium transparency. Another one theory is arrangement of stromal lamellae. It is explained by Morris theory and Goldman's theory. According to Morris theory, the corneal transparency is a consequence of crystalline lattice arrangement of collagen fibrils within the stromal lamellae, and that light scattered by the individual fibrils of uniform diameter is cancelled by destructive interference with scattering of light from the adjacent areas. Therefore, light is scattered only in forward direction. Here we can see the lattice pattern arrangement of the stromal laminae as explained by Morris theory. Another theory is theory of Goldman et al. He postulated that cornea is transparent because the fibrils are small in relationship to the light and they do not interfere with the light transmission unless they are larger than the one half of wavelength of light. This theory is confirmed by founding of legs, which are divide of collagen fibers with the dimension greater than 2000 angstrom in swollen non-transparent human cornea. Third important factor which maintains the corneal transparency is corneal vasculature. The cornea is avascular except for small loops which invade the periphery for about 1 mm. However, various disease processes of the cornea may be associated with corneal vascularization. Another one factor is corneal hydration. The normal cornea maintains itself in a state of relative dehydration, which is essential for maintenance of corneal transparency. All these factors, along with the endothelial pump, the endothelium contains high concentration, the endothelial pump, which is mainly sodium potassium ATPases. In endothelial cells, the extracellular space, the sodium potassium ATPases in exchange for the potassium. The endothelium also contains high concentration of carbonic anhydrase, which forms the bicarbonate from metabolic CO2 ion. The bicarbonate ion diffuses down its concentration gradient into the extracellular space or across the membrane associated with chloride and bicarbonate exchanger. Bicarbonate also appears to enter the cells via sodium bicarbonate cotransporter, and the intracellular pH may be regulated by basal sodium hydrogen exchanger. Here we can see at the Descemet's membrane and endothelium side there is exchange of sodium potassium ATPases as well as bicarbonate ions through the aqueous humor. The drug permeability across cornea. The corneal epithelium and endothelium is lipophilic and are crossed readily by non-polar, that means lipid soluble substances. The stroma is being hydrophilic, so it is easily crossed by polar, that means water soluble compound. So drug should be amphipathic so that it can easily penetrate across the cornea. The ionic forms of drugs. Only non-ionized drug can penetrate through the epithelium as it is lipid soluble, and ionized drug can pass through the stroma as it is water soluble. Therefore, a fluorescein is a negatively charged ion, so it cannot penetrate the intact epithelium, and this property forms the basis of fluorescein dietase. The rest of details of corneal diseases and the important structures which we should be knowing in the cornea will be discussed in next chapters. Thank you so much for your patience listening.